Hello, my name is Ari Silver, and I'm an undergrad here at UF. I am currently a junior, and I'm majoring in microbiology and cell sciences. And a fun fact about me is I'm a vegetarian who absolutely hates salads. It's kind of a conundrum, I understand that, but it is what it is. So today I'll be discussing circular RNA, CDR1AS, and how it may be a potential treatment for glioblastoma. And glioblastoma is one of the most aggressive cancers. It originates in the brain, and my lab actually studies GBM, or glioblastoma. So that's one of the reasons why I picked this article, because this cancer is so deadly, and it's important to know more about it. So let's start off with some basics or some background about some of the mechanisms of how cancer may spread or grow in the brain. So we see here in response to cell stress, such as DNA damage or hypoxia, P53 is activated, and when activated, it causes the activation of genes which cause apoptosis and cell cycle arrest. So essentially, if a cell gets damaged or mutated, or it suffers hypoxia, some sort of stress, P53 is activated and it tells the cell, I'm sorry, buddy, but you gotta die. And the cell commits suicide. But inactivation of this vital protein can facilitate tumorigenesis because cells that may be mutated are not able to commit suicide or end their progenies. So they continue to multiply, 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 and that's cancer. And many types of cancer have a loss of function mutation in the TP53 gene, which codes for P53. However, GBM is very atypical. Most GBM tumors have intact TP53, so they're still producing P53, which means that the protein itself is being inactivated, not the gene. So here we have a bigger picture of the pathway of P53, and we see that one of the genes that P53 stimulates actually codes for MDM2, and MDM2 forms a negative feedback loop with P53. MDM2 inhibits transcriptional activity of P53, and it promotes its ubiquitination and degradation through proteasomes. So MDM2 is important in the body because too much P53 is not a good thing. So it helps control and regulate the amount of P53 in the body. However, GBM hijacks this cycle, overexpresses MDM2, which causes a decreased function of P53 at the protein level. And this is when disaster strikes. This is when things get way out of hand. However, there is an unsung hero to this story, and the hero's name is CDR1AS. It's a circular RNA, and it's able to bind to the P53 MDM2 complex to destabilize it and activate the P53. So as we see in this graphic, CDR1AS comes in with his cape like Superman. It's ready to save the day. It binds to P53 through P53's DNA binding domain and ejects MDM2. So now P53 is able to carry out its vital functions once again. And the paper I'm going to be looking at today questions whether inactivation of CD1AS should promote tumorigenesis in GBM, or vice versa, whether upregulation of CD1AS will prevent tumor growth. Here we have a graph showing how different glioma tumor grades correspond with CDR1AS expression. So we have here on the lower end WHO's second grade glioma, grade 2, has relatively high levels of CDR1AS, but as we travel down to WHO's grade 4 glioma, or glioblastoma, at this point, we see significant downregulation of CDR1AS. So this shows us that already in GBM's microenvironment, we're seeing downregulation of CDR1AS to promote tumor growth. Here we see that CDR1AS has high P53 enrichment, which shows binding between the RNA and the protein. In this figure, we see a heat map showing levels of p53 binding to CDR1AS, and since it's red, it corresponds to a very high level of binding between them, while we have a control protein IgG, which does not bind CDR1AS, and it has very low levels of expression. Similarly, when we look at CDR1AS's place in the chromosome X, we see P53 binds to CDR1AS in its chromosomal area very highly as compared to IgG, which does not bind very highly at all to its locus. Here's a really cool image showing immunofluorescent staining of P53 compared to RNA fish of CDR1AS and DAPI, which is a nuclear stain. And we see here the merged image, and we see how P53 and CDR1AS overlap very well. Here's a zoom in of the merged image, which tells us that CDR1AS and P53 are geospatially linked within the tumor. This shows us that if you'll find one, most likely you'll find the other one. CDR1AS actually promotes P53 expression at the protein and not at the mRNA level. So as we discussed previously, the gene for P53, TP53, is not affected by CDR1AS. However, the protein P53 is affected, and this graph shows this. So as we can see in the figure on the left, increasing levels of CDR1AS in a cancer stem line, U87MG, shows increasing levels of P53 as CDR1AS increases. However, in the same cancer stem line, we see that increasing levels of CDR1AS, 500, 1000, 1500, shows no effect on the levels of the gene TP53. So this shows, as we stated earlier, how GBM with CDR1AS is affected at the protein P53 and not the gene TP53 level. Moving on, we see that CDR1AS is able to extend P53's half-life in the same GBM cells. And we used an immunoblot to analyze this, and we see that when exposed to CHX, which prevents protein synthesis, we see that in the control, P53 degrades relatively quickly, but when exposed to CDR1AS, P53 degrades much more slowly. We also see MDM2 has the same effect as P53. However, you must realize that P53 stimulates MDM2's production, so we expect more MDM2 to begin with, as P53 levels were higher before the experiment began. Also, CDR1AS prevents the function of MDM2 to degrade P53, so increased levels of MDM2 should not be that big of a deal if we have increased levels of CDR1AS, which explains why P53's levels continue to be elevated. Here's a quantification of this. We see the control has significantly decreased levels of P53, and we see that when CDR1AS is present, we see much higher levels of P53 for longer periods of time, which shows that CDR1AS prevents ubiquitination and degradation of P53. And here is more evidence directly showing that CDR1AS prevents P53's ubiquitination. And we have Si, CDR1AS 1 and 2, different isoforms. And we know that Si in this paper means knockdown. So whenever we see Si, we know it means a knockdown CDR1AS. So if we're knocking down CDR1AS, we have less of it, so we would expect increased ubiquitination of p53 in these areas where it is knocked down. And that's exactly what we see in this immunoblot of p53 ubiquitination in CDR1AS knockdown GBM cells. And these cells were transfected with ubiquitin and MDM2 plasmids and exposed to MG133. So we see here, when exposed to knockdown SICDR1AS, isoforms 1 and 2, we see increased ubiquitination, but in areas without the knockdown with normal expression of CDR1AS, we see a lot less ubiquitination, which shows us that CDR1AS is playing a direct effect in preventing p53 from being ubiquitinated and degraded. And in this MG132, for this experiment, it prevents proteasomes from degrading ubiquitinated p53, which allows this experiment to mean anything, to have significant results. Moving on, CDR1AS binds to p53's DNA binding domain, and we realized this with this experiment where we showed that we exposed CDR1AS to different mutated versions of p53. So the first MYC p53 is just normal full-length p53. ND2 means deletion of two N-terminal domains. CD1 means deletion of two C-terminal domains. And MD1 means deletion of the DNA binding domain. And we see that level of p53 binding to CDR1AS is significantly decreased when p53's DNA binding domain is deleted, which leads us to conclude that CDR1AS binds to p53's DNA binding domain. And through this binding, it stabilizes p53, disrupting the p53 MDM2 complex. So it's important to show one of the main goals of this paper is CDR1AS potential as a therapeutic option. So now we're seeing if CDR1AS impedes gliomagenesis, or the growth of glioma cells in vitro. And we have SINC, which is the control, and SICDR1AS1 and 2, which is knockdown CDR1AS in both of these. And what we see is that the U87MG GBM, or glioma cells, grow significantly more when CDR1AS is knocked down. And this is quantified here by colony number. We see that the control has a lot less colonies than when CDR1AS is knocked down, which is exactly what we would expect. And this same trend holds true in vivo in mice models, which is very exciting. And what this shows is that the tumor volume is significantly greater when CDR1AS is knocked down as compared to a control with normal levels of CDR1AS. And the same experiment has the survival curve. It's the Kaplan-Meier survival curve, and we see that in the control, the mice survive for much longer than in the CDR1AS knockdown mice. So this paper has many very interesting implications. The most important being that it shows that CDR1AS can actually bind proteins. Previously, we only thought that CDR1AS could act as a miRNA sponge to affect gliomagenesis. So the previous model shows CDR1AS acting as a sponge to collect miRNAs or sequester them so that they could not be used to cause mRNA degradation. However, now we're realizing that they can be used to actually bind proteins, which is a huge step. And this adds an entirely new layer of control over tumorigenesis to CDR1AS. Future direction. This paper also has a lot of potential for future direction. One of them being we can learn more about how CDR1AS affects other targets throughout the body. So we just discovered it can bind proteins. What other proteins can it bind? We can elucidate the other mechanisms to disrupt the p53 MDM2 complex. Now that we see that this complex is so vital to gliomogenesis, what other molecules can disrupt this complex? If the logic being if CDR1AS can do it, surely other RNAs or proteins may also be able to disrupt the complex. And lastly, to develop therapeutics to upregulate CDR1AS, this is to me the most important because this can be translated to clinical trials for drugs that can upregulate CDR1AS and prevent gliomogenesis or slow down the growth of tumors. So, to conclude, who knew that an RNA or a circular RNA could do so much or be so powerful in terms of binding to proteins and slowing down gliomogenesis or the growth of glioblastoma in the brain? Targeting p53 MDM2 has been a very long-standing goal for decades of researchers, and we're finally finding an RNA that's able to accomplish just that and disrupt such a vital or crucial complex for glioblastoma growth. And I will end with a potential caveat is that treatment of upregulating CDR1AS is only effective if functional p53 is produced by the tumor cells. So many tumors have non-functional p53 being produced or mutations in TP53. But if p53 is functional, then this treatment can work. And luckily for us, many glioblastoma tumors have functional p53. So to conclude, this is extremely exciting research, and I'm very excited to see the future potential of CDR1AS in treatments for glioblastoma. Thank you so much, and here are my references.